Good day and good evening, everyone. Thank you for having me here. It's a great honor today to join you for this major launch event and to also share with you my view on the challenges for developing and commercializing a microRNA-based vaccine. We all know, so next slide. We all know that biomarkers are playing an increasingly important role today for successful drug development and for patient care. From understanding disease mechanism, disease status, monitoring disease progressions, predicting and optimizing treatment, and monitoring clinical outcomes. Many biomarkers can be developed into diagnostic tests for various applications, including risk identification, like BRCA1, BRCA2, those type of biomarkers, or for screening, again, BRCA1, BRCA2 is a good example. Diagnosis, prognosis, predictive, or dynamic monitoring whether disease relapse resistant or minimal residual disease. Although many biomarkers has been successful into diagnostic tests, majority, I would say probably 99.9% of biomarkers never Hello? Oh, okay. Okay. So next slide, please. So these figures on the slide are a little bit old, but was based on my research, a very quick PubMed research. So the graph on the left shows that summarize the number of published articles in PubMed that reported microRNA or messenger RNA or methylation-based biomarker assays. In the gray bars represent the total articles reported as the assay for each type of biomarker. The orange bar are articles mentioned or presented signatures. Signatures meaning a panel of microRNA or a panel of methylation markers or messenger RNA. And the blue bar are the articles actually present a AUC curve that describe the performance of the signatures. So these were from data from PubMed that I searched 2000 to 2020. So based on that quick search, you can see anywhere from 100 to 40,000 people have been published for this different type of biomarker with or without AUC. However, on the left, look at R. First, I want to apologize. There's some error in the graph. The numbers are correct, but the graph wasn't updated. I first did the analysis in 2016. And so the graph was done in 2016, but after that in this May, 2020 May, I went back to the ESI, look at how many messenger RNA-based biomarker signature versus methylation versus microRNA-based signature that have been approved by US FDA, not including any in other countries. So you can see there's very, very few that have been approved by FDA. And the first gene expression-based test was actually approved back in 2007. And as of May 2020, only 6 messenger RNA gene expression-based tests have been approved by US FDA. I think 2 of the 6 were related to colon cancer, Cologuard as well as Epicolon, I believe. And then not one single microRNA-based test has been approved by US FDA. So I want to take this opportunity to congratulate Myrex that having the first microRNA-based test approved by Singapore government. I think that's one major milestone. As Frank mentioned, there are 2 LDT launched in US, which is one is for thyroid cancer, the other one was actually the very first one by Rosetta Genomics. And there are several in Asia, but none in US that was approved by US FDA. And one thing I also want to emphasize, US FDA approval does not guarantee successful commercialization. Must be able to demonstrate clinical utility. The reason I want to mention that is if you look at my figure, the graph on the right-hand side, there's one star that I say including gene search. Gene search was actually the first gene expression test that was approved by FDA. Shortly after it was launched, it got taken out of the market because the workflow is very difficult and also there's limited clinical utility. So this is the one lesson learned by many IVD industry. Having FDA approval does not guarantee commercial success. The next slide. So why is it so hard? So much effort on biomarker identification, study, assay, all the assay performance being reported, but very few get to the regulatory approval and get to the market. So why is that? So since today we are talking about microRNA standard launch in Singapore, so I'm using microRNA for lung cancer as an example. The top graph on the X are each one of the microRNA, each individual microRNA. The Y-axis are the number of times in the article search that I carried out back in 2016. So the data will be very different today. But back then, just to show you, only one microRNA that been reported 4 times out of the 16 articles, only one microRNA that reported 4 times. So 4 out of 16. So that tells you there's some kind of reproducibility issue. Majority of the microRNA reported that are highly associated with lung cancer, they have been reported only 1 time, and they were never reproduced by other articles or other researchers. You look at the bottom, on the other hand, all of these microRNAs that have been reported for various different cancers, the number one microRNA that is most cited by lung cancer on the top is microRNA 21, which is also reported 10 times, which also reported in 10 different cancers. So therefore, that's another issue, specificity. So I'm using cancer as an example. Out of that 19 different cancers, one microRNA is reported in 10 different cancers. So you have reproducibility issue, you have specificity issue. So why is that? Well, as Frank mentioned, oftentimes these microRNAs are very small, very difficult to quantitate. So the sensitivity and specificity of the assay analytical performance is extremely important. So next slide, please. What are the key challenges? So we already talked about the analytical variability, and Frank mentioned that. But in reality, when you develop any biomarker assay, there are many types of different variability that we need to overcome. The first variability is the biological variability. When you take a tube of blood or a piece of tissue, what you see in that tube of blood and what you see in that piece of tissue is a snapshot. The biomarkers are constantly changing, whether you are running just now or whether you are stopping fasting overnight or whether you're just being very dehydrated or have a big, fatty meal, that changes your biomarker. Everything you do changes your biomarker. So what you measure, regardless of what tool you use, it's a snapshot. So we have to understand the biological variability. That's something we can not control very well, but we have to understand it very well. But on top of that, the sample quality, how you collect the sample, how you store the sample, how you pack the sample, those are pre-analytical variability. And then on top of that, you have different tools, different reagents, different detection methods. All of these added on to the variability. These are analytical variability. And after you finish all that, you have lot-to-lot reagent. So the reagent provider, the manufacturer, who manufacturing these reagents, they have process variability, they have raw material variability, etc., and operator variability during manufacturing. So all these variabilities stacking up. And then by the time you have 5%, 10% here, then your total variability is what? You don't know, right? So that's the reason why it is that difficult to develop a very robust any biomarker assay, especially multivariant, whether it's microRNA or messenger RNA. So next slide, please. So the key is that we need to understand and control the various different types and the source of the variability. So to develop and commercialize the novel biomarker-based diagnostic test, it's very important. First of all, you have to have very robust biomarker discovery and development, and always start with the end in mind. So make sure your analytical tools are robust, control the sample quality and pre-analytical variability, understand the biological variability. For example, do you need to have, collect the sample after fasting or not? If it's a urine sample, is that first meal or not? Whatever. So you need to understand the biological variability. And then, of course, very importantly, you have to develop your biomarker, discover and develop your biomarker with appropriate samples and study designs, including using the right intended population for the intended use. That's what I meant with the end in mind, and statistic power, and understand what are the potential bias and confounders. And then following that, you need to make sure you have very robust performance for the diagnostic assay for the intended use. So very comprehensive analytical validation would be required, and of course, ideally you would follow the requirement and guideline to make sure everything works well. And then prospective validation, again, follow the guideline with the intended population for intended use and appropriate study design with statistical power. And finally, very importantly, you have to demonstrate clinical utility to help drive guideline adoption and to help drive reimbursement. If you don't have guideline adoption and you don't have a strong clinical utility, you cannot achieve reimbursement. If you cannot achieve reimbursement, then the global adoption would be pretty difficult. So next slide, please. So in terms of microRNA-based diagnostic, it's particularly challenging, but at the same time, it also offers a great opportunity. So if you look at the microRNA, it is every single one of microRNA, so shown as this figure. Actually, blocking this. Okay, so the microRNAs, this is one of the review paper that are specifically looking at microRNA in the database with a set of was to analyze the network, the pathway, and mapping all the microRNA pathways together for primary hypertension disease. The minimum common microRNAs for each one of the microRNA annotations that and the minimum of the target gene that are type or related are 1,000. So you have anywhere from 600 to almost 6,000 genes that are inside it. And if you look at the gene network, it's quite complicated. A lot of crosstalks and a lot of loops. So that tells you this is a vast hypertrophy of microRNA activity. So one single microRNA can regulate many networks, many pathways. And so it is therefore very difficult to actually pinpoint which microRNA for a specific disease diagnosis. So the lack of specificity on the pathological status of any given microRNA, and that's the major challenge. However, because these microRNAs are, as their role serving as a regulator, they can provide deep insight into the complexity of the gene regulation, molecular pathway, crosstalks, feedback loops, and the cascades, etc., and provide us an opportunity to better understand what are the interactions between the pathways, and those molecular mechanisms that underlie the disease onset and progression, and as well as all those associated comorbidities. So I think that microRNA really because of its complexity, because of its regulatory network, it provides us a tool to better understand in depth and broadly the disease, the phenotype, and the associated mechanism. Also, because there are many microRNA, several microRNA will regulate one path, one microRNA will regulate many different paths. So therefore, the opportunity is that you could potentially identify microRNAs, a panel of microRNAs, to detect a highly heterogeneous disease, because this panel of microRNA may be able to shed light and cover all the disease regulations and mechanisms that will help diagnose the disease, whether at early or at advanced stage. So next slide. So looking forward, given that microRNA is much more stable molecule, and it provides a very good tool for us to better understand disease mechanism and potentially developing microRNA-based signature for diagnosis. I'm very hopeful, looking forward that this standard that we just launched will actually help us not only to develop robust diagnostic tools for various different clinical applications, but also will help develop insights for drug discovery, drug development, and to achieve microRNA-based precision medicine. So with that, I want to conclude my talk, and I want to congratulate everyone who has worked on this standard and who has made contributions to this standard. So thank you again.